Okay, in this short video, we just want to talk about the fundamentals of mitosis. So in this course, we're not going to emphasize the different stages of mitosis as much. We're going to focus on basically what's the point of mitosis and what needs to be accomplished and basically how is that done. So as we will probably remember from the other videos, the point of mitosis is to duplicate the cell, to make two genetically identical cells from a single cell. And so our major tasks then are to duplicate the DNA and to divide. So it's important that we need to divide right. Okay, so different chromosomes have different genes. So we need to make sure that all of our cells have all the genes, so they have all the chromosomes, that we don't end up with two different cells. We can't just divide it at random. So if we think about this, so we start out with a haploid cell, and then all the chromosomes are duplicated, so now we have two copies of each. So we can't just sort of divide the cell in half, because that could leave us with, here we have two blues and no greens, and here we have two greens and no blues. And so this is a good time to emphasize that chromosomes are all profoundly different. So here on the left we have chromosome 1 and chromosome 17 of humans. And so what we're looking at here is, sorry, that's not legible. Each one of these is a different gene that's been associated with some sort of a condition. So this list of genes here is totally different than this list of genes. So it's not just like you could substitute chromosome 1 for chromosome 17. Right? When we draw things in terms of lines, well, these kind of look like they're the same thing just with different colors. So remember, it's important to remember that a chromosome is made up of individual genes which have totally distinct functions. So having three chromosome 1s but only one chromosome 17 is going to cause major differences in cell and organismal function. So indeed, if we just look at, if we think about it, so here's chromosome 21, these are individual genes where it's been shown that lacking just one of these genes can cause substantial medical problems, can cause substantial disease. And so if we were to just delete or fail to properly inherit these cells, to segregate these cells, this chromosome into different cells, that would be a big problem, right? We'd be missing all of these genes. If we didn't get this chromosome correctly divided into the two daughter cells, we'd have two cells with a lot of problems. And so now we can just learn how the cell basically fixes this problem or deals with this difficulty. So the first thing is after duplication that the chromosomes stay attached. So here I've shown the blue chromosome duplicating, and now the orange and the green ones, and they're going to stay attached at a single point. So now these cells, these two sister chromosomes are attached at a point called a centromere, and they're going to line up along what's called the metaphase plate, basically the center of this cell, the center of the nucleus. And then they're going to be attached to these two processes on either side called centrosomes and microtubules. Okay, so we've got the centrosomes. They're basically the things that are going to tug. They're going to pull on either side. So this one's going to be pulling to the left. This one's going to be pulling to the right. So, and by pulling, they're going to use these microtubules that they're attached to the chromosomes so that they're going to be pulling apart. And basically when everything is lined up properly, they are going to separate, and now we have a single set of chromosomes on the left and a single set of chromosomes on the right. Now we can divide the cell, actually divide the surface of the cell and the cytoplasm and all the components, and now we have two descendant cells. That's